and we are ready for Professor Davi. I'm very excited about his talk. He's a professor at the University of Paris, and he has been one of the founders, as I mentioned, of European Biomed 2, now called EuroClonality, who standardized analysis of antigen receptor genes for clonality assessment. So Professor Davi is going to talk mainly about standardized NGS-based assays and protocols for clonality assessment, for minimal residual disease monitoring, and repertoire analysis applied specifically to lymphoid malignancies, so in hemato-oncology. Thank you very much, Professor David, for being here today with us, and we're very much looking forward to your talk.
Hello, everybody. Thank you for this introduction, and thank you for the invitation to this extremely interesting session. And I'm glad to be to acknowledge your community. I used to be a clinician, and in France, hematology can be either clinical or biological, and I'm head of the Department of Biological Hematology. And as you know, the diagnostic of malignant hematology condition is based on cellular hematology with cytology and flow cytometry, and genomics, looking both at chromosome and genes. And as Professor Ross has explained before, and thank you for this very nice presentation, everything is under regulatory control, which is the norm, the ISO 15189, and the national body that controls that is the COFRAC in France. Now, in contrast to the US, where I think having accreditation has been going on for years and years, it's fairly new, at least in France, and started about, well, let's say 10, 15 years ago. It's a very slow process. Not everything that we do is accredited, and I can tell you that our lab has probably about 60, 70% of its tests accredited. So we've been used, and it's, of course, the situation is changing, but to work with reagents and tests which were not accredited. But it's slowly and surely changing. And I fully agree with what has been said before, that going through this process is long, cumbersome, and takes a lot of energy. Now, what we do in the molecular diagnostic for malignancies is, for a long time, we were just looking at single targets. Professor Ross spoke of the BCR-ABL fusion, which can be detected both in chromosome and gene, and which is very useful for diagnostic and response to treatment, minimal residual disease assessment. So this is probably one of the oldest tests that was developed in the lab, and we're just in the process of, actually, we have gone through the accreditation. Same thing, single target like TP53 alterations in chronic lymphocytic leukemia can be assessed both by chromosome and genes. And this is important as a prognostic indicator, but more and more to decide which treatment will be given. Of course, as has been shown before, we more and more use NGS looking at multiple targets. We have a much smaller panel than Dr. Ross, only 60 genes, but of course it's going to move. And this is useful for identifying tumor variants, tumor subtypes, and in the future for giving the right drug to the right patient, which is called the precision medicine. So besides accreditation, when you use that kind of test, you need something special. This is just somatic genetic alterations, so there's no specific request. However, especially with large panel, you may find constitutional abnormalities, and then you need the patient consent, and also you need to be qualified for that kind of genetics. So whenever we find those variants which happen, we collaborate with our geneticists, and we give them everything to be done, and eventually having a consultation for the patient. Now, just to put things, so this is true for all kind of cancer. And I would just show you this picture, this epidemiological survey, which was done a few years ago. Hematological malignancies represent about 10% of all cancers in France. And 2/3 of them are lymphoid origin, which means that beside those genes, cancer genes that we can search for, the antigen receptor are excellent targets. And I don't need to this audience to explain you why, but with a huge potential diversity, each cell has its own arrangement, and it's really molecular identity of a cell. And to go back in history, that was the idea 40 years ago now to use this as diagnostic biomarkers. And shortly after the discovery of the VDJ recombination that was used for diagnostic of lymphoma. Of course, at that time, this was used with a 1,000 blot, which was a fairly long technique, but robust. And for many years, this is what we used. And when I did my postdoc, and this is a picture that was taken from a work I did in Jeff's class lab, we could really identify rearrangements in, here it is an acute lymphoblastic leukemia, where it's a tumor from precursor lymphoid cells, which still undergo VDJ rearrangement during the evolution of the disease. Of course, in the 90s, there was a lot of development on PCR, focusing on the CDR3, because as you can see on this slide, in a normal population, every lymphocyte has a different CDR3 sequence and length, whereas in a tumor, and most of tumors arise from mature B cell or T cell tumors, the rearrangement is fixed and all the tumor cells share the same clonal rearrangement. So at the end of the 90s, there was a project which was called the Biomet2. It was financed by Europe, was one of these Biomet2 concerted actions, which had the goal of standardizing PCR-based studies for diagnostic of clonality in lymphoid proliferations. And this was there again, as you've seen in the presentation before, a large collaboration study involving 48 institutions. There were molecular labs, but also pathology labs. So we designed and the protocols, the primers and the protocols, and there was a central production of primers, and we tested these primers on a collection of samples. There was about 100 of them, which had been very well characterized by pathologists and molecular using 1,000 blot as a gold standard. And there was an initial testing by a few laboratories, and then there was a further testing by 10 laboratories. So we designed assays for all genes, all targets, all kind of rearrangements, the VDG and the DJ for IGH and beta, and all light chain, and also TR gamma and delta. At the time, the analysis was heteroduplex or gene scanning, which led to this paper that some of you may know, which was really the proof that the collaboration could provide a very robust test for clonality assessment. And I believe the Biomet2 primers and PCR assays are still used by many laboratories. And there was a work package to assess those tests in collections of B cell malignancy and T cell malignancy, with the idea that if you want to do clonality, you often have to analyze several targets, especially in B cell malignancies, because of the somatic hypermutation that could hamper the amplification of targets. And also what we did, which a lot of people found useful, because clonality, and still after doing that for many years, I find that this is one of the most difficult molecular tests for interpretation. So we provided guidelines for interpretation and reporting of clonality. So beside clonality, there were parallel efforts for minimal residual disease with the same spirit. And another European consortium called AIRR-C tried to, or attempted to standardize the IGHV mutational status in CLL. I will come back to that in a minute. So these European networks provide assay development and standardization, so protocol guidelines, but they also have external quality assessment sessions, workshops to train new investigators. And there's also online support when somebody in the lab has problem, either doing an experiment or interpretation. So are there any specific regulatory issues for that part? I would say no. And this is why it came really easily in all our labs. Clonality is just PCR and fragment analysis. And for minimal residual disease and IGHV mutational status, you need to sequence, which was done before by Sanger sequencing, and use a tool like GTV-Quest to identify the genes. So about 10 years ago, with the advance of NGS, we decided to see whether we could do the same thing for clonality, minimal residual disease, and IGHV mutational status. And there again, it was exactly like PCR. We did not invent anything. There's a lot of assays, a lot of tests which have been published, but we just wanted to have something that could be robust and used in many clinical laboratories. So I will start just before that. So this is so a number of labs coming either from EuroClonality, from EuroMRD, or from AIRR-C joined together. And this was the same kind of organization that was the previous Biomed2. And there were a work package that were designed and leaders for each of them for primer design. There was a specific work package for bioinformatics, because this is obviously something extremely important. And then there were application core projects to clonality, minimal residual disease, and quote-unquote repertoire. So I was with Kostas Stamatopoulos in charge of trying to set up something for IGHV mutational status in CLL. For those who are not familiar, in this disease, which is a proliferation of mature B cells, and for a long time it was noted that cells could have either unmutated or genes, IGHV genes, or mutated. So with the possibility there were two different diseases. And in 1999, two papers showed that the load of somatic mutation in the IGHV genes had a dramatic impact on the clinical evolution. So with a threshold at 2%, which is still the one which is used, and when cells carry PCR with less than 10%, it's much more aggressive disease. This has been confirmed. This is a study we've done in in 7,000 patients. So it's a very strong and fixed biomarker. And it's also a predictive marker, and patients who have mutated genes with no other genomic alterations have a very good and maybe cured by the standard immunochemotherapy. So there are more and more reasons to perform this test in a clinical situation. So traditionally, what you just need to do is having the sequence, count the mutations compared to the germline, and just then determine if the percentage of identity is below or above 98%. So this can be done from genomic DNA or cDNA. The importance is to have the full size of the V gene, which means having primer in the leader peptide region. So we decided to set up an assay starting from genomic DNA. And we spent a lot of time designing the primers on the L2 part of the leader. So the assay includes 23 primers and one J primer. It's a one-step PCR, And because of the length of the PCR product, we use a MySeq platform with 600 cycles. Very important is to have what I call user-friendly bioinformatics. Most of us in the clinical labs are not bioinformatic experts. It's difficult to have access to that kind of people. So we needed something that was easy to use for us. So two pipelines were developed. One on purpose by the team of Nikos Dazentas, which is called AIRR-Interrogate, and one which is pre-existed, which is Vigil, led by the French team of Mathieu Giraud. I would say that one of the big assets of this software is they're quick. They offer a graphic interface, and they are linked to other tools, so you can go from them. Here, an example for Vigil. Some of you may know this is a CLL with biallelic rearrangement. You just need to click, so you have the gene scan type view. You have also the VGene and the JGene, which are used. You have the clonotype frequency. You can just click on IMGVT, and you have exactly the same thing, the same result that one would have with Sanger. Same display, or same type of display with AIRR-Interrogate. It's a bit more complex, but you get the same kind of information. So initially, we used 125 CLL, which had been sequenced by Sanger. And we used our assay to see whether we had the same result. And the answer was yes, 96% of the cases. And we had 3 cases where we could see the clonotype, but it was a minor one. And there were 2 cases where there was a long stretch of poly G in the CDR3, which made the identification of the gene, of the rearrangement, a bit difficult. Then we started to use it prospectively in the cases that we had in the lab. And as you can see, in almost 90% of cases, we could assess the IGHV mutational status. There were about 10% of cases where we could not, either because they had no PCR product linked to mutations, either in the 5' or the 3' primers. We also made an inter-laboratory reproducibility test. We sent 24 DNA samples to 4 expert labs from the AIRR group with all the reagents and the protocols. And as you can see, except for one lab which failed on 2 cases, we got exactly the same sequence, which I mean 100% identical in all labs. And it's also, to some extent, a quantitative, and you can see they had the same result. As you can see in this graph, interestingly, I forgot to say that the mutational status is done only on the productive rearrangement. And as you can see, there were some cases where the clonotype was very weak. And these were, in fact, cases where there were biallelic rearrangements, where the productive rearrangement was amplified much less than the unproductive one. So we decided to optimize the protocol. And for that, we designed an assay where we cloned rearrangement from CLL in plasmids. And we had 47 rearrangements, each of them having a different V gene. And we made then an equimolar mix of this plasmid. So we tested our initial mix. It was clear that in theory, because there's roughly 50 plasmid, which each of them should be sequenced and represent 2% of the whole. It was clear that there were some plasmid, some genes which were under-amplified or over-amplified. So we spent a lot of time optimizing the assay, but so after many attempts, we got something which is not perfect, but much better. And as you can see, for example, the range of frequency between 1 and 3% was 27, and now it's 32. And we have very few genes which are above, are either over-amplified or under-amplified. And this is what it does under those 2 cases where we had unbalance. So before optimization and after optimization, you see that it's much better, and it's even more clear in this case. So we use now this optimized protocol, and we were able to amplified from a case of from 650 CLL. Now the rate of success is 93%. And there are cases where either it's a failure or there are minor clones, which may be problematic to interpret. So for that, we went to a complementary test as working on cDNA, where then it can use primers on the L1 part, which are less mutated, but which are on the genomic DNA far from the L2 because of the intron, but on the cDNA much closer, so easier to sequence, and also can use a constant primer regions. Initially, we designed for C mu and C gamma, and we found cases that were C alpha and C delta, so we added those two primers. We checked on 100 cases that all these primers were amplified, were able to amplify all V genes and J genes, which was the case, except for what is in the red here, cases which didn't have cDNA available. But for two of them, we know that they were amplifiable on genomic DNA. And the other are very rarely, this is the frequency of IGHV usage in CLL, which, as you can see, is very unbalanced. So the ones which we were not able, which we didn't test, are very rarely used. And then we went back and took 59 cases from our routine test, either that had genomic DNA failure or which needed confirmation because of minor clone or really unbalanced between two alleles or oligoclonality. And in all of them, we were able to confirm a productive rearrangement and to assess the mutational status. There again, we also did an inter-laboratory reproducibility test as exactly with the same condition, and it worked very well, with a difference that with cDNA we observed a quantitative differences which were a bit more marked than with genomic DNA, though that may be linked to the cDNA degradation. So we believe that this test is almost ready to go for an accreditation, but of course we will need to do everything that has been described by the previous speaker. Our colleagues from Rotterdam used this assay for evaluation of MRD. So it's you needed to add a few things, and they compared to what they used in the lab, which is either digital PCR and quantitative PCR. And you can see that's a very good correlation. The sensitivity can be very high, and if you use enough DNA, you can have sensitivity up to 10 to the minus 5. And it showed also to have a clinical utility because in the protocol where MRD has been set up with a threshold at 10 to the minus 4, going further could identify patients which had an intermediate survival, even free, progression-free survival between those who were really negative and those who were 10 to the minus 4. Another approach was done for MRD in acute lymphoblastic leukemia. And as I've shown you from the thousand plot, these are tumors where there are often multiple genes and multiple targets which are rearranged. So another work package took care of designing an assay, which is a bit different here. It's a two-step PCR, where all the targets are amplified. And then in the second step, and the barcodes are adapted and sequencing adapters, so you can distinguish the type of patient, the patients, and the type of test which has been done. Importantly, there is always a polyclonal target, which is a mix of polyclonal cells from lymph node, tonsils, and blood, which can monitor primary performance, and which is very important for MRD, is have an in-tube control, which is a spike in of 40 cell equivalent of known cell lines, which have 24 well-defined IG rearrangement, and which help to normalize the assay and convert reads to clonotypic reads to cells. And there was a multi-center assay validation comparing NGS versus traditional PCR for target identification. And this was done by 5 labs on 50 cases of acute lymphoblastic leukemia, which showed a very good concordance, 78%, with a tendency that NGS would identify more targets than conventional Sanger. And then this assay was used in protocols, in the retrospective protocols, and it showed in the top paper that yes, going to a low level of MRD can be detected by a low and a high throughput, and that NGS is a better discrimination for true MRD positivity in acute lymphoblastic leukemia. Now going to clonality, this was again another work package, and here there was drawn by a pathologist. So as a pathology working very often on FFPE material with the DNA is degraded or not always in optimal quality, so often short fragments, we needed short amplicons. So there was a design using 3 targets, FR3, DJ, and kappa and kappa deleting elements. And you can see the traditional image by GenSkin and GenScan and the equivalent in NGS. AIRR-S interrogate has a special feature where if you use this assay, you can have automatically the results on GenScan and clonotype frequency, which for all targets, for all 4 targets, which is very helpful and easy to use in the clinical lab. There was a validation study that was done on more than 200 samples in 4 laboratories, which showed a very good interlaboratory concordance, and as has been shown for PCR and GenScan, the use of complementarity for immunoglobulin targets in lymphoma. So are we going to use this and is that going to replace clonality? I'm not sure, but I can see some indications, some cases where it's really useful because PCR GenScan is really simple and very robust and cheap. But this is 2 examples here for detection of minor clones, Hodgkin lymphoma, and as you know, probably the tumor is a B-cell tumor, but the clone is only a minor clone surrounded by a lot of inflammatory cells. So quite often by GenScan, you don't see anything. However, in this case, with NGS, you can clearly identify a clonotype on the heavy chain and the light chain. So, and I'm sorry for the poor quality of the bottom of the slide, but comparing Biomed2 classical PCR GenScan and NGS assay to detect clones in both fresh frozen or FFPE tissue, there was a superiority of the NGS approach. So for that kind of cases, I do believe that NGS is extremely interesting. And this is a case from our lab on TR gamma. This is a patient that was, and where I believe that NGS is really helpful. This is a patient which had a lymphoma that you can see a big clone in the skin, in the blood. I'm sorry, there's a mistake. That should be 2018. And he was treated, and then he was followed up. And in 2021, there was this small peak at the size where was the clonal peak before. And the question is, that really the tumor or not? And same thing in the blood. So we could not really confirm just by using conventional method, but with NGS, because you have the sequence of the clonotype, you can really see that in both of these diagnostic, obviously, you can see the sequence of the clonotype, and you can show that they have exactly the same sequence in this small peak here in the skin of 2020, but the clonotype is very different in the blood of 2021, you do not detect the the tumor clone. Now, just I would go briefly that we also, there was another workshop where we can use a different type of assay. Everything that I've shown you so far is amplicon-based, but one of the group decided to use capture. And the reason is that you can do at the same time for tumors looking at oncogenic variants and immunogenic. So they put some probes to cover all the antigen receptor genes. But also, which is interesting, to detect translocations with regions for probes switch and genes which are often translocate to those immunoglobulin or TR genes. So for the bioinformatics, you can either recognize by one of part of the pipelines the sequence, but also you can use the copy number alteration to show that the intergenic between the D and the J here and the V and the J here has been deleted, which can be helpful in case of somatic mutation where it's not easy to recognize the VDJ recombination. There was a validation study which was done by 7 labs, and this showed a very good ability to detect B-cell malignancy, T-cell malignancy. The limit of detection was 5%, and as I told you, you can detect at the same time a copy number alteration and single nucleotide variants. And here's a map of the variants which were detected in this collection of tumors. And there's about 70 genes which are on the panel. So in terms of regulatory issue, I will just go briefly in this because it will be discussed. I think what is important is for bioinformatics. And so far, VGL, as one of the 2 tools that we used, has made the effort. So there are, as you may know, there are 2 versions of the VGL, one which is a public server, which is free. It's more like a research, it's for researchers only. And there is a hospital version. And they went through all their accreditation, quote unquote, process. So they will follow their ISO 27001, and they are they followed their compliance with the healthcare data hosting regulation, GPDR, and the French certification. And we can use VGL for accreditation, which we will do in the near future. Now, a big challenge for us, and I'm sure it will be discussed, is IVDR. A lot of the tests that we do are homemade. Even if we follow exactly the recommendations, but for cost reason, we avoid to buy kits. With the incoming IVDR, it will be absolutely impossible unless one can show that the test we perform is better than the one which is on the market, which means that we will have to buy a lot of kits, which of course will be very expensive, especially because the industry will have to charge more than research RUO only kits, and even more than the CE IVD, what we use in Europe for those kits. So this is a complex situation, but I'm sure that will be discussed later on. So future for immunogenomics, I'm sure it's bright. In oncohematology, of course, there's those 3 main applications, but I can see and I listed a lot of applications which may be addressed. And of course, immunomonitoring in other medical conditions is something which I'm sure will be developed in the next coming years. So I just wanted to thank you for your attention and thank all the people who participated in this work.
Thank you so much, Professor Davi. That was a very nice presentation, very interesting indeed, with lots of application, lots of standardization efforts that have been performed in time. So it was very interesting. We don't have much time, but I'll see if there is one question in the audience before we go to the next speaker, and then we leave the rest of the questions for the panel discussion. Is there a question from the audience? Felix?
Sorry, yeah, that was fascinating. And my question may be kind of odd, but I mean, the AIR community is one of the goals is to make these data shareable and easily comparable. And here you're presenting, I mean, you're a researcher and a clinician, and of course you want to be able to compare these data. But I often wonder, I mean, will it, if you do you think a person who's mostly a clinician, are they going to just want to know, okay, it hits the 10 to the -4, clone level or not? Or are they ever going to want to be able to compare other data sets and look for those, like you had those multiple samples and you were looking for those, minimal clones and stuff? Does that make sense what I'm saying? How much do we want to build systems where these clinicians are going to be able to easily compare to other patient sets and control sets?
Are you talking of minimal residual disease? If I understand.
Or yeah, sure. Or see, these other assays. How many is the clinician just going to want to know? Okay, it hits these parameters. Just tell me that. Or do you imagine that clinicians will want to be able to explore other data sets and compare their patients to a bunch of other data sets?
I think that, yeah. Well, there are two questions. Either you have a clinical question, which is can be very, okay, a patient has an enlarged lymph node, you send it to a pathologist, the pathologist, it might be lymphoma or not, but I'm not sure. And then you want to know if that's a clonal population. So you want to have a test that will be able to tell you, yes, there's a clonal population. And same, minimal residual disease is becoming something more and more important because it's a way to address the response to treatment. And especially in cases where it would take a long time to have the response in terms of survival, if you have an assay that can tell you that the molecular disappearance is linked to survival, then you can just measure that and have the answer. So there is a big pressure for MRD assessment with very sensitive technique. So this is just a really fairly standard, really clinical question. Now, a more interesting question is scientifically. Okay, that's more for immunogenetics, a repertoire study. Is leukemia, such type of leukemia, are they really specific? If I sequence it in my lab, is that the same thing in your lab? And is that the same thing from those kind of patients? Is that what the question? And then
I think we'll chip a little bit in the question, and I'll add my part to Felix's question, and then we see that because it's interesting one. We can leave this also for the panel discussion later, but maybe we conclude before we move on. Basically, I think what is being asked is, the clinician. So if you would imagine a traditional clinician that is not a researcher or a scientist on the side at night, would they need to know all of this validation, all of these assays, clones, bioinformatics, or would they need only to know, okay, the specific clone that will indicate a minimal residual disease or an MRD that is above this threshold, 4% or whatever, will indicate me that I would need to choose treatment A versus treatment B. So what is the information? You mentioned it user-friendly, but what is the information that the clinician would need to know? Is this all the science that we have been able to appreciate during your presentation in time, in 20, 30 years? Or do they need like basically the threshold to say, okay, my patient has above 4%. That's it. Like I know now what I will treat him because the minimal residual disease is this or that. What do you think? Would clinicians appreciate the refined science behind, or do they actually need to get to the result?
Well, they want to get to the result, but it depends what you ask for. For diagnostic, you want to be sure, and you set a threshold, for example, 1%. You say everything above 1% I can consider as a clone. I will tell you there's a clone, it might be malignant. For minimal residual disease, there is evidence, and I'm sorry, maybe I didn't go. There was one slide where I showed that the deeper you go, the better you are. So technology is important because with NGS, you might be able to go very deep, and that might be a difference.
That will tell you that the disease is still there, even at a low clonality. I got you.
Absolutely.
Thank you so much for your presentation. We'll have a panel discussion. That's why I'm extremely excited, Felix. We bring it further to the panel discussion. We'll address everything.